Welcome, students, to this PharmaEd Explainer. I'm your professor, and today we're going to simplify the absolute complexities of medicinal chemistry for your exams. If you've ever felt completely overwhelmed by looking at endless chemical structures, well, don't worry. Today we're going to connect chemistry, pharmacology, and microbiology into one clear, beautiful, logical story. All right, Section 1, Core Concepts of SAR. Let's start from the very foundation of how we actually design medicines. So SAR simply means how modifying a chemical structure literally changes a drug's biological action. Think of it like this. If I change a functional group, does the drug get more potent, less toxic? Does it absorb better? That's SAR. The Pharmacophore, on the other hand, is the absolute essential three-dimensional shape required for binding. It's not the whole molecule, no, just the crucial parts, like hydrogen bond donors or aromatic rings that actually turn the lock. Concept clear?
Are you noting this down? You should be. These 4 factors dictate if your drug was fit the biological receptor perfectly. First up, electronic effects. Groups like halogens can pull or push electron density, which alters how tightly the drug binds. Second, steric effects. Imagine trying to fit a bulky key into a tiny lock. It won't work, right? A bulky group might help a drug fit snugly into a hydrophobic pocket, or it might just block binding entirely. Third, lipophilicity. It's a delicate balance. A drug has to be lipid-soluble enough to cross cell membranes, but water-soluble enough to travel in the blood. Finally, stereochemistry. Receptors are 3D, so the exact 3D stereoisomer of your drug completely dictates its potency and selectivity.
Moving on to Section 2, Antibacterial Classes and SAR. Let's take these fundamental chemistry concepts and apply them directly to the real world. Now, this table is very, very important for your university and competitive exams. Notice how we categorize antibacterial drugs strictly by their cellular target. This is the whole principle of selective toxicity. We want to destroy the bacteria without harming the human host. So we target the cell wall with beta-lactams, protein synthesis with aminoglycosides, nucleic acids with fluoroquinolones, and folate metabolism with sulfonamides. Let's look a bit closer at these structures. Okay, the beta-lactam rule is probably the most critical SAR point you will learn today. That 4-membered beta-lactam ring, it is absolutely essential to bind to penicillin binding proteins, PBPs, and stop cell wall synthesis. If you break this ring, your antibiotic is completely dead. Useless. The free carboxyl group is also strictly required to interact with the target. And that acyl amino side chain, well, we modify that side chain to protect the drug from stomach acid, to expand its spectrum, or to shield it from those destructive beta-lactamase enzymes. Real quick, before we move inside the bacterial cell to look at our next targets, please like, share, and subscribe to PharmaEd for more pharmacy lectures. Your support really helps us bring more high-yield concepts right to your screen. All right, moving inside the cell now, you need to memorize these targets and structural features. Aminoglycosides, like gentamicin, are highly polar, water-soluble molecules with multiple amino sugars that hit the 30S ribosomal subunit. Tetracyclines, with their very characteristic 4-ring fuse system, they also target the 30S ribosome. Macrolides, like erythromycin, have this massive macrocyclic lactone ring and hit the larger 50S subunit. And then we have the fluoroquinolones. Their SAR requires a 3-carboxylic acid group and a 4-keto group to successfully block DNA gyrase and topoisomerase 4. That effectively halts bacterial DNA replication. Now, what's really fascinating here is the strategy of sequential blockade. You see, bacteria must synthesize their own folate to survive. Sulfonamides structurally look almost exactly like PABA, para-aminobenzoic acid. So the bacteria get confused, they take up the sulfomethoxazole instead, and boom, the first enzyme, dihydroopterate synthase, is blocked. Then trimethoprim steps in to inhibit the very next step, dihydrofolate reductase. It's a brilliant 1-2 punch in medicinal chemistry that gives us beautifully enhanced antibacterial activity. Let's shift our focus a bit now. Section 3, Antifungals and Resistance. This is a massive modern challenge we're facing. Fungi are tricky, beta. They're eukaryotes, which means they're closer to human cells than bacteria are. But they use a molecule called ergosterol in their cell membranes instead of human cholesterol. And that right there is our target. Polyenes, like amphotericin B, are these large molecules that bind directly to ergosterol, physically tearing holes in the fungal membrane. Azoles, like fluconazole, work a bit differently by inhibiting the actual biosynthesis of ergosterol. And allylamines, like terbinafine, go even further upstream to inhibit an enzyme called squalene epoxide. But the microbes fight back, beta. They always do. And this is exactly why medicinal chemistry never stops.
Bacteria develop enzymes like beta-lactamases just to chop up our antibiotics. They mutate their binding sites so our drugs no longer fit the pharmacophore. They even build efflux pumps to literally spit the drug right back out of the cell. Crazy, right? So as medicinal chemists, we must constantly design structural modifications, maybe adding steric bulk to avoid enzymes or tweaking lipophilicity to evade those efflux pumps, all just to retain the drug's activity.
All right, close your notebooks. It's time for your final exam. Section 4, GPAT MCQ test. I've selected 5 high-yield mock questions based on what we just covered to test your memory right now. Let's see who gets full marks. Question 1. What does SAR primarily describe? Read the options on your screen. I'm giving you 3, 2, 1. The correct answer is B, chemical structure and biological activity. Remember, structure dictates absolutely everything in medicinal chemistry. You change the structure, you change the biological response. Moving to question 2, and we talked about this extensively. Which structural feature is essential for the antibacterial activity of penicillins? What is the absolute heart of the molecule? Lock in your answers in 3, 2, 1. Correct. It is B, the beta-lactam ring. Without this 4-membered cyclic amide, the drug just cannot bind to penicillin binding proteins, and there is absolutely zero cell wall inhibition. Question 3. Fluoroquinolones primarily interfere with what? Think about the quinolone nucleus and where it acts inside the cell. I'll give you a hint. It happens deep in the nucleus of the bacteria. 3, 2, 1. The answer is B, DNA replication. Those 3 carboxylic and 4 keto groups on fluoroquinolones are absolutely essential for targeting DNA gyrase and topoisomerase 4. Question 4. Trimethoprim primarily inhibits what? Recall that beautiful 1-2 punch, the sequential blockade we discussed earlier. 3, 2, 1. Excellent. If you got B, dihydrofolate reductase. Remember, sulfamethoxazole hits dihydroparoate synthase first, and then trimethoprim comes in and finishes the job in the folate pathway. All right, final question for top marks today. Let's see if you are really paying attention to the target differences. Ergosterol is an important component of the what? Where do we find it? 3, 2, 1. The answer is B, the fungal cell membrane. It's a key component that sets it apart from bacterial and human targets. If you understand this flow, from chemical structure to physical-chemical properties, to target interaction, to mechanism, and finally biological activity, you really don't need to rote memorize. You simply follow the logic. As microbial resistance keeps evolving and these superbugs get stronger and stronger, my final question to you is this. Armed with the power of medicinal chemistry, what radical new molecule will you design to save the world? Keep studying hard, Beta. I'll see you in the next explainer.